This lecture overview is for the second part of chapter 13 on host defenses, so host defense part 2. So we talked through the initial part of this chapter 13, looking at the lymphatic system and how that relates to the lymphocytes and where they migrate to, the primary lymphoid organ where T and B cells are maturing, and then the secondary lymphoid organ where T and B cells are essentially hanging out to be activated. So we're going to talk through some of that stuff today on these slides on host defense, and I'm going to minimize my video. So in this on these sets of slides, we'll finish up this whole concept of the activation and response of your T cells and your B cells. There are some slides at the end on immunity, which we'll cover here, and then on vaccination or looking at immunization, which since I'm recording this, I might as well include those ones too. There are key concepts for this one, just as we mentioned before, essentially the whole process from development to response for both B and T cells. And for vaccines, we may or may not touch on the vaccine controversy, so we'll get to that if we have time for. Okay. So this should look familiar. We've talked through this already a bit. This is the B and T cell line. So again, both of them derive from that original pluripotent stem cell or hematopoietic stem cell in the bone marrow. They that stem cell differentiates into lymphocytic precursor stem cell, which then can further differentiate into either a B cell line or a T cell line. Now, because of their shared origin, it actually allows us to group these guys together under the same shared lineage, and they do have some similarities, which we'll see, which we saw last time when we were looking at B and T cell receptors, and that unique variability that's provided with that, the specificity of the antigen binding site for both B and T cells. There are also some receptors that are shared for lymphocytes, which are helpful in evaluations of total blood cell count for lymphocytes, etc. We won't go into any of those details, but just so you know, they do have some shared lineage there. We remember that B cells are developed in the bone marrow and then stay in the bone marrow for their differentiation. This is also important. The differentiation part is important in the development of that specific B cell receptor, which, if you recall, we called the immunoglobulin class D, which is usually on the surface of naive B cells. We'll do a brief reminder when we get to that point on immunoglobulins and how they look in their different regions. These guys hang out or finish their negative selection in the bone marrow, and then as long as they're not self-reactive, they leave the bone marrow, which is the primary lymphoid organ, and then they circulate through the blood and migrate to the lymph nodes or spleen or other secondary lymphoid organ. There's a whole bunch of different examples of these or regions throughout the body of these, essentially these collection sites for our naive lymphocytes. For T cells, if you recall, they again begin their development in the bone marrow, and then they move to the thymus, where they complete their development and differentiation, gaining their unique antigen specificity with their T cell receptors. Remember that T cells also have those co-receptors, CD4 and CD8, and those are important in being able to recognize MHC class 1 and class 2. And that requires a positive selection. So T cells in the thymus, before they're able to leave here, they undergo both negative selection so that any self-reactive T cell is deleted, and positive selection so that the MHC class 1 and class 2 are both appropriately recognized by either CD8 or CD4 co-receptors. Once all of that stuff is completed and the T cells are essentially approved, then they leave that secondary or that primary lymphoid organ, the thymus, and then they circulate through blood to get into the same area as we mentioned for B cells, either the lymph nodes or the spleen, and somewhere in the body. Once the lymphocytes reach the secondary lymphoid organ, they only have a short window of time, a couple of weeks maybe, before they either self-destruct or become activated. So there's a short window, there's constant turnover of our lymphocytes in there. We mentioned at the very end of the last video about antigen presentation. So all of the stuff in the beginning is in the absence of antigen. And then in order for activation and then clonal expansion in effector cells to be produced, then it's an antigen dependent response. So this is where we have our MHC class 2 presentation for antigen presenting cells. This would be dendritic cells if this is the first time around, or macrophages, or even B cells, which we'll talk about today in these slides here. This occurs in the secondary lymphoid organ, this antigen processing presentation. And then once, if the appropriate, or if there is a perfect match with the T cell receptor, then we'll have T cell activation and then clonal expansion, producing lots of different types of clones. And we'll see this process here. This is called cell-mediated immunity. We also will watch the development of a B cell response or humoral immunity response. And we're going to focus on the T dependent, which means T cells go first in their activation. They become effector cells, and then those effector cells can then go downstream and activate B cells. There are other ways that B cells can get activated, but this is one very classic pathway and one that we'll focus on here. We mentioned already that the cell-mediated immune response is with T cells, and usually when we're talking about cell-mediated immunity, we are primarily talking about cytotoxic T cell response, but there are other types of T cells that we'll talk about today. For humoral immunity, we're talking about the B cell response, and this is really just antibody production. There are 5 different classes of antibodies that we mentioned before, and so today we'll differentiate between those different classes and talk about what the function of each of those classes are in the different antibody responses. So before we get there, actually I want to take a little segue and go over to our whiteboard and just talk through those different types of responses. And hopefully this will give you a little idea as to the different, when the body would do one type of response versus the other. So let's give a couple of examples here. Over here on the side, I'm going to just draw some epithelial cells, maybe like small intestine or colon, or small intestine. Small intestine. Okay. And over here, another one. Okay, so these are just epithelium. Epithelium. And let's just say that this is in the intestine. There are some organisms, like E. coli, for example, that have toxins that do the damage, but they themselves are extracellular, so they stay outside of the cell when they colonize. So extracellular pathogens, that means they stay outside of the cell, they're not invasive, versus intracellular pathogen. I said intercellular pathogens. And these guys get inside the cell, right? So classic example, of course, would be a virus causing an intracellular or invasive colonization or infection. So there's a difference, obviously, between the ability for our body to respond to pathogens that get inside the cell and cause damage, or pathogens that stay outside the cell, usually release toxins or cause some kind of a immune response that does the damage here. Remember that all pathogens, of course, do damage. So there's a different way for our body to respond to whether we have an extracellular pathogen or an intracellular pathogen. For the extracellular guys, even if they can get past our epithelium and they get into blood, move through circulation, they're still not going to invade a cell. So to combat extracellular pathogens, usually we mount a humoral response. And if you recall what we just said, this means antibodies. antibodies, which also means we are talking about a B cell response. To mount a humoral immune response, there are helper T cells. So there are some cells that help with this one. Th2 is really good at activating B cells and really pushing for this response. And also T follicular helper cells, which we'll talk through both of these classes in just a minute. So both of these ones are helper cells to really push for B cell production, to develop those antibodies to combat that extracellular pathogen. Antibodies will then come outside and they essentially, they'll just bind to the surface of our pathogens and neutralize them, or there's other functions of antibodies, but that's kind of the main thing here, is that they can go and bind to the surface of these organisms and really stop them in their tracks. However, an antibody response is not going to be as effective for intracellular pathogens. Now, when we have viruses, we do mount a humoral immune response, but there's another type of response that's also really, really great here, and this is where we would get cell-mediated response. Specifically, our cytotoxic T cells. Which we call TC, cytotoxic T cells. And these guys, their job is to go around and find any self-infected cell and essentially cause them to self-destruct, right? So it's a fantastic mechanism we have in place to help get rid of any intracellular pathogens. There are also other T helper cells involved in the cell-mediated immune response. We have a boost in macrophages' ability to combat anything that's on the inside of them. So this would be a Th1 response, but usually those responses actually induce greater inflammatory events. So we'll just stick with this one here, the cytotoxic T cells. T helpers are also important in getting our cytotoxic T cells to get going, but we'll look at that also in a minute. I just really wanted to make sure you guys are understanding that when we're discussing our different types of lymphocyte responses, that it really depends on the nature of the pathogen, what type of response is going to be required and boosted for the development of that response. So we have been talking through these 4 stages of development. We already discussed lymphocyte development differentiation. We discussed real briefly, with the aid of my children, the presentation of antigen on the surface of our antigen presenting cells. And now we're going to talk today through the challenge of B and T lymphocytes, and then also what occurs after the challenge of those lymphocytes. So the antibody response or humoral immune response for B cell activated B cells, and then cell-mediated immunity for the activation of T cells. So I know that your book doesn't talk through the organization of a lymph node, but really we're going to go back to our discussion of the lymph node and talk through all of this stuff. And I told you before, we're going to do a lot of whiteboard writing for this chapter. So bear with me, let's go back to the whiteboard. So we're going to talk through first T cell activation, and then we'll talk through cell-mediated immune response, and then we'll go through the B cell activation, and that really is going to get us through the majority of this chapter. But to do so, let's go back over to our whiteboard. Okay, I'm going to clear this one. And we're going to redraw our lymph node. I'm probably going to have to draw this a few times, but let's see if I can make it kind of big. Okay, so if you recall, we have this lymph node here. We have all these draining lymphatic vessels here coming in. These are our afferent vessels. So we have incoming lymph, right? That didn't work. Okay. Forget that one. Okay. So this is coming from, or this is lymph that's coming in to, oh my gosh, I'm sorry guys. Lymph that's coming in to the lymph node, and we're drawing a lymph node here. From the tissue, right? And so we're going to remind ourselves there is some kind of injury that occurred, right? Whether this is an infected tissue or damaged tissue. And this is like from chapter 12 where we talked about injury. And we remember from chapter 12 that when injury occurs, then there is an inflammatory response. The primary responders are neutrophils, and then the late-term responders are macrophages and dendritic cells. So dendritic cells arrive at that injured tissue. They engulf or phagocytose whatever is there. And then I mentioned to you before that as soon as these guys begin the process of phagocytosis, they hightail it back to the lymphatic system. Let me draw them in green, I guess. So these dendritic cells will be coming through the lymph and then drain into our lymph nodes, in addition to dendritic cells that are now antigen presenting cell. So they're going to be fully loaded, and I'll put some receptors on the surface here. If you recall, we have MHC class 2 receptors on antigen presenting cells, but I'm going to kind of confuse you a little bit and tell you that we also have MHC class 1 on these antigen presenting cells too. So here's a whole bunch of antigens that these guys picked up, and they're going to be bringing all of those antigens presented on the surface of their on their MHC class 2 receptors and be bringing them into this lymph node. In addition, there's also, so we have our APCs, but there's also free floating antigen. And sometimes free, that means they're not bound, right? And sometimes you might actually have the whole pathogen, but we're gonna kind of stick it to this, that we have these incoming dendritic cells, and then we also have free antigen. It's sort of all just floating in here in the lymph. Since I have the ability to draw a bunch of different colors, let's keep our dendritic cells a different color. Okay. So this stuff is all coming into the lymph node, and the lymph node has these afferent vessels, the stuff that's bringing lymph into the lymph node. The lymph node has these things called sinuses. And just like your sinuses essentially, you know, draw in and drain things out from your nose to your, whatever, to your respiratory tract. I don't know if that's a good comparison, but these sinuses are sort of where things kind of migrate through. Now the sinuses also have these drainage regions that sort of migrate all the way through the lymph node. And so stuff that's coming in, it'll sort of go in that direction, and it'll move down the sinuses, and it can also drain down into these drainage regions. And the relevance of this is that there are different kinds of zones. So down here is our T cell zone, right? Where we have all of our incoming T cells that are naive, they're just kind of hanging out and waiting to go. We have both T helper cells here. And these are naive. And we also have cytotoxic T cells that are hanging out here. TC. And they're also naive. Okay, and then if you recall, our T cells have some key features on them, right? They all have their unique T cell receptor. And that variable region provides them, you know, variation from one cell to the other. And same thing on these T cells. These cytotoxic T cells still have T cell receptor. And then we have our co-receptors. So let's draw our CD4 co-receptor here on these naive T helper cells, right? CD4. And for our cytotoxic T cells, we have CD8 co-receptor here. Okay, CD8 and CD4 co-receptors. I guess I'll just, I'm gonna redraw these ones over here too. Right, so these guys have CD4 co-receptor. They have that CD3 anchoring receptor, and they have their T cell receptor. And then our cytotoxic T cells are our CD8 T cells, right? They also have their CD4, I'm sorry, their T cell receptor, and they have that CD8. Oops. CD8 co-receptor, also with their anchoring CD3s. All right, so this is what we're talking about, these two types of cells. We talked about these ones before and how their co-receptor provides them specificity as to which type of MHC compound and therefore which type of cell they can bind to and become activated. So in addition to these this T cell zone, we recall there are these B cell zones essentially. These are the follicles or the follicles, which can have germinal centers on the inside. So these are follicle, which is where the B cells are. And once you have proliferation, so once you have a B cell that's activated, they form these things called germinal centers on the inside. And so you'll actually see these B cells kind of aggregate in one particular area. So these are the follicles, and the germinal centers are part of the follicles that can be formed upon B cell activation. So these are all the B cell zones are sort of all hanging out on the outside region, and the T cell zone is here on the inside. So this organization becomes really important as we begin talking about the step-by-step activation of our immune system, of our adaptive immune system. Okay, so we have our injured tissue out here. Our dendritic cells are getting fully loaded with antigen, and they start to migrate towards the lymph node or secondary lymphoid organ through the lymphatic tissue, right? The lymphatic vessel. And as they enter into this afferent vessel, both free antigen and these antigen presenting cells are coming into the sinuses, and they begin to drain through the sinuses here. And I'm not going to talk to you about all of these different regions here, but essentially we have the drainage almost like a coffee pot percolator, right? So these sinuses can actually drain through this lymph node and essentially allow these dendritic cells, and I'm going to tell you right now that I'm on the side, if you ever take an advanced immunology class, you'll find that I'm not telling you the full truth about this, but we're going to try to keep it as simple as possible. And I think this will do a good job of sort of talking to you about how this whole process occurs. So this APC is going to come in and essentially through a very particular way will can essentially get down into this T cell zone and to go and try to find a T cell, starting with T helper cells, but also cytotoxic T cells, that has a perfect match to one of its presenting antigens, right? So this dendritic cell will essentially be down here and kind of moving through. So here's our APC, and it's presenting antigen on its surface. And it's looking for a perfect match. So it's just going to kind of migrate through this area and try to find a T cell, either T helper cell or cytotoxic T cell, that has this perfect interaction here, this perfect match between the variable region of the T cell receptor and that antigen it's presenting. If a perfect match is made, then this causes the activation of that matched T cell, and it can then lead to something called clonal expansion. So let's look at this kind of step-by-step process. Now, I do want to take a second and let you know that I'm gonna add something here. These antigen-presenting cells, these APCs, they present antigen on actually both MHC class 2 and MHC class 1. So they have both MHC class 2 and they have MHC class 1. MHC class 1, which remember that we talked about from our last set of slides that MHC class 1 is always presenting self-antigen. And if it's presenting anything foreign, it's usually a sign that that cell is infected. Now this is the only exception to the rule. For APCs only, in secondary lymphoid organ, I'm gonna just do 2, secondary lymphoid organ, APCs can do something called cross-present or cross-presentation on, well, let's say on 4, my gosh, excuse me just a second. Okay, cross-present, where they put a foreign antigen on MHC class 1, right? This is unique because always if you have foreign antigen on MHC class 1, that usually always means that that cell is infected or damaged and it causes the destruction of the cell. But only in the secondary lymphoid organs, antigen-presenting cells may cross-present foreign antigen on MHC class 1, right? So they will be able to activate both naive cytotoxic T cells in this regard and also classically naive T helper cells, right? So, and classically on MHC class 2. So this allows for activation an APC to activate both both T helper cells and cytotoxic T cells. So let's try to draw a picture, and I'll just show you this concept of cross-presentation here on APCs. It doesn't usually happen this way, but I'll try to draw it anyway. Let me see if I can maybe make a new, all right, just a second. All right, let's try to redraw this. Essentially what we're going to do here is draw out cross-presentation of our antigen presenting cell to activate both T helper cell and cytotoxic T cell. Okay. T cell activation. So let's draw out our antigen presenting cell. And again, I'm going to draw this in one way. Know that I'm just going to show you one APC, a dendritic cell, activating both cytotoxic T cell and T helper cell. So here's our APC, and this is going to be a cross presenting. All right. So let's draw our, oops, I think we're doing it in green. MHC, nope, that didn't work. I don't like that color either. Okay. So this is MHC class 2 receptor. It's going to present a foreign antigen. And so let's say that this is in the lymph node. Where this APC essentially has already undergone the whole process of phagocytosis and pulling out particular antigens, and now they're presenting those antigens all over the surface of their cell, right? Here's MHC class 2 all over. And all of these guys have various types of antigens on them. And they're essentially just looking around to try to find any matching T cell or a T cell that matches these particular antigens. So let's try over here first, our T helper cell. And remember, this is naive. That means that it has developed its receptors, it has left the primary lymphoid organ and has come to hang out here in the secondary lymphoid organ. And so we have our T cell receptor, and let's just say that this is a perfect match. So here is T cell receptor with the variable region and the constant region. And let's say that this is a perfect match here. And then we also have our CD4 co-receptor. What color did I draw that in? I don't remember. All right, so here is our CD4 co-receptor that interacts with MHC class 2. Okay, so this is one interaction. We looked at this one before where we see if there's a perfect match between the foreign antigen and the T cell receptor. And in addition, if the CD4 co-receptor can bind to and recognize MHC class 2, this causes activation of our T helper cells. So actually, let's continue drawing this one out. Okay, so what we just saw here is a perfect interaction between MHC class 2 being recognized by that CD4 co-receptor on the T helper cell. And this is APC. And then there's also that perfect match with that antigen, foreign antigen, is matched perfectly with that T cell receptor. So this leads to activation. And although activation, there are a lot of things that are happening during activation. And don't forget, we also have CD3 co-receptors here. But for our sake, the first thing that we're going to mention here, this is the nucleus. This interaction will lead to a signal cascade down to the nucleus, and it will change gene expression in the nucleus. All right, here's our DNA. To make the cell do something different. So activation, and let's kind of do this step by step. Step number one essentially is this binding here, and that would lead to activation. And activation for our T helper cells, we're going to say, is the production of a growth factor that's really important called interleukin 2. And this is a lymphocyte growth factor. And this also will lead to, so that would be number one, or the first thing that's done here. Activation is the production of interleukin 2. So here now we have secretion of interleukin 2. And this acts, this is a cytokine that's important for essentially building up a lymphocyte response, both B and T cell response in the lymph node, but it's also important in self-activation. So interleukin 2 is produced, and also interleukin 2 receptor. So this interleukin 2 acts as a growth factor for any lymphocyte that's there, but also for itself. So this is the interleukin 2 receptor here. So interleukin 2 will come and bind back in on itself, right? So it acts in some ways as an autocrine, right? Those are hormones that can activate the cell that they are being produced from, but these are cytokines, right? So it's a little bit of a different concept here. These cytokines, these interleukin 2s, go out into the lymph node, essentially in the surrounding area, but also are important in activating its own cell. So here's interleukin 2 receptor. This leads to a second activation event. So the binding and engaging of interleukin 2 receptor will also trigger secondary events here. And in this case, this leads to something called clonal expansion, which is the rapid proliferation or replication of this activated cell to produce three types of, or to produce two major classes of cells to produce into effector cells. The effector cells are the ones that actually go out and do the business. Effector cells and memory cells. And these guys are the ones that hang out for a long period of time. Okay, so clonal expansion is the rapid proliferation of this guy into these effector cells, which are the ones that go and do something, and into memory cells, which hang out for a long period of time. So let's go through this whole process again. Here we have an antigen presenting cell, and right now we're just focused on presentation to our T helper CD4 positive naive T cells. So these guys are hanging out in the secondary lymphoid organ. Our antigen presenting cell comes in and sort of starts to percolate through the T cell zones. Here is where it has MHC class 2 presenting foreign antigen. If this perfect match is made here between the foreign antigen and the T cell receptor, and this is a CD4 positive T helper cell that recognizes that MHC class 2 complex, This leads then to activation. And I will say that there are other cytokines that are released from this antigen presenting cell that actually tell the cell what type of effector cell to be produced, okay? Let me pause just for a second. So let's just put some numbers to this. Okay, so if this interaction here, where MHC class 2 presenting for an antigen is recognized by CD4 co-receptor and a perfect match is made between the variable region of the T cell receptor and the foreign antigen, this essentially would be step 1, is this engagement here, right? So 1 is this interaction. This leads to this whole cascade reaction, the activation of this T helper cell. So we're just going to say all of this stuff here would be step number 2, would be activation. This is the production of both interleukin 2 and the interleukin 2 receptor. The engagement of the interleukin 2 receptor with interleukin 2 itself would be 3, okay? And this leads to clonal expansion, which essentially is 4, okay? 1 is the whole receptor engagement up here leads to activation, which is 2, interleukin 2 and interleukin 2 receptor are produced. And the engagement of those two together again leads to another trigger event that can change everything in this T helper cell and leads to rapid expansion of this one particular cell, replication into clones that are either effector cells, which go and do something, or memory cells, which hang out for a long period of time. Okay, so let's keep this here. We're going to talk about these effector cells that are produced in just a second. But I want to also mention, because we have it up here already, that this whole thing happens not just with T helper cells in the lymph node, but also the activation of T cytotoxic cells here, okay? So in this regard, let's draw our cytotoxic T cells over here too. I don't remember our color scheme from our other slide. Oh, we had that in orange. Okay. So remember, this is still in the T cell zone. And here we have our cytotoxic T cells, right? These are CD8 positive cytotoxic T cells, and these are also naive cells. These guys have the same T cell receptor. Well, the same basic structure. And they also have specificity for their T cell receptor at the top there. And instead of CD4, these guys now have CD8. CD8 co-receptor. And we recall CD8 co-receptor binds to and recognizes MHC class 1. And still, we must remember that this is all about antigen presentation. So now here's a different type of antigen. Let's just say it could be the same, it could be different. And here's that foreign antigen. And this is cross-presentation right here. It means that these guys can present on both MHC class 2 and MHC class 1. So they are presenter cells. That's what they do. That's their whole function. And in the lymph node, uniquely, they can present on both, whoops, they can present on both MHC class 2 and MHC class 1. Right, so here's MHC class 1. All right. So antigen presenting cells can cross-present in the lymph node on both MHC class 2 and on MHC class 1. And so here we have an example of this presentation of antigen. And same as before, right? If on the APC, versus the TC. On the APC, we have MHC class 1 that's recognized by the CD8 co-receptor on the cytotoxic T cell. We have foreign antigen on the APC that's being recognized by the T cell receptor on the cytotoxic T cell. And this then leads to activation. So very similar to what we saw before. And again, if we're putting our little numbers to it, we can say number 1 here is this engagement here, APC to cytotoxic T cell through this cross-presentation of foreign antigen on MHC class 1. This then leads to activation, and whereas this one is the production of interleukin-2 and interleukin-2 receptor, here again we're going to kind of keep it simple, and we're going to say the activation in this regard for cytotoxic T cells leads to the production of interleukin-2 receptor. All right, so here's the nucleus. There's the cascade reaction that changes the nucleus and makes these guys produce interleukin-2 receptor. And I'll put them over here too. Interleukin-2 receptor. Interleukin-2 receptor then can bind any interleukin-2 that has been made from neighboring T helper naive activated T helper cells. So here's IL-2 from activated TH cell. And so now we can have this interleukin-2 receptor bound by interleukin-2, and this leads to another signal event, which then leads to clonal expansion. Let's put our numbers to these things. So here we had this receptor engagement leads to activation, which is interleukin-2 production and interleukin-2 receptor. Then we said that after interleukin-2 is produced, if it binds back to that receptor, this would be step number 3. And then that receptor engagement leads to another change, which would lead to the rapid proliferation and the production of effector cells and memory cells for clonal expansion. So here we have, again, receptor engagement for cytotoxic T cells that are naive with antigen-presenting cells cross-presenting foreign antigen on MHC class 1. This leads to activation, which is going to be step 2, which is the production. Where's our hot pink? There we go. Step 2, which is the production of interleukin-2 receptor. And again, I am just madly simplifying this. There are lots of other steps that are involved in the activation and differentiation of these guys, but we're going to keep it this simple. Step 2 would be activation, which is the production of interleukin-2 receptor. This interleukin-2 receptor can then bind to interleukin-2 from activated T helper cells, which is important to note that there really does need to be these T helper cells activated first and foremost before anything else downstream can really get going. If this receptor engagement occurs, this would be step number 3. Is this receptor engagement, which leads to clonal expansion, which is step 4, right? And clonal expansion, just as before, leads to the production of effector cells, which are the ones that go out and do something, and the production of memory cells. And actually, I will note that in every one of these events where a cell is made into, is expanded through clonal expansion, there's always regulatory cells that are made. Regulatory cells that are made. So I'll add that here too. Regulatory cells, regulatory cells. memory cells, and effector cells. I know this is a lot of information, but hopefully you can just take this step by step all the way through both activation of T helper cells and the production of effector cells here, and the activation of cytotoxic T cells and their production of effector cells. So let's go down into here and look at the effector cells that are made from T helper cells and the effector cells that are made from cytotoxic T cells. But first I'll go back to our slides. Okay, so activation, remember, occurs in the, sorry, this should say secondary lymphoid organ. I haven't fixed that yet for whatever reason. Occurs in secondary lymphoid organs, and that would be something like lymph node, spleen, etc. So here we have the presentation of by an antigen presenting cell on both MHC class 2 and MHC class 1. And don't forget that this concept is this cross presentation here, but this is that complex that we looked at before. Here's CD3 co-receptor, here's CD4 co-receptor that binds to and recognizes MHC class 2 that's presenting a foreign antigen, and here's that T cell receptor that has to engage with that foreign antigen there. The binding causes T helper cells to release interleukin-2 growth factor for both T helper cells and cytotoxic T cells. And this interleukin-2 binding back to itself promotes this concept of clonal expansion. Clonal expansion is just making tons and tons of copies of the exact same cell, but they're differentiated. We call them terminally differentiated. That means that these effector cells, once they're made, they're going to do something and then they're going to die. Versus memory cells, which are incompletely differentiated cells that last for a very long period of time. So for this concept again of clonal expansion, just so you see, multiplication or massive replication is what we're talking about here. Multiplication equals replication of the exact same cell, right? So there's the original activated T helper cell, for example, and it'll divide into making copies of itself. Okay, and these, the majority of them will be effector cells. That means that they go and they do something, right? These are the most numerous progeny. They're actually larger in size from their undifferentiated counterparts, the naive cells, but they're only short-lived. They go and do something. Effector cells means functional. They have some kind of function. They go out and do some kind of function. Versus memory cells, these ones are incompletely differentiated, so they still retain the same antigen specificity, retain antigen specificity. Actually, that's important for all of these. For every single type of cell that's produced from this original cell, all of these things have the same antigen specificity. All of them do, right? That means that all of them have the same exact T cell receptor as the original cell did in the first place. And this is how we have this highly specific response in the adaptive immune system, is because of this retention of antigen specificity, which means the same T cell receptor. And so even for these memory cells that hang out for a long period of time, they can react with the exact same antigen in the future, and their steps towards activation is much, much faster. So actually within minutes to hours of having that antigen, same exact guy come into the body, they will react and they will form a response. But let's look now at the actual effector cells for CD4 T cells. So there's quite a few variations of the types of effector cells that can be produced from an activated CD4 T cell. And all of these things will have a different type of function in the body. And this will all depend upon what the initial antigen presenting cell is essentially communicating to its bound T cell as to which type of T cell will be made. So I'll tell you, this is actually a short list. There are a lot more types of T helper cells, effector cells that we know of, but we're going to essentially keep it simple and just look at this short list here. So classically, T helper 1 versus T helper 2, these are two different types of T helper cells that have two different functions altogether. So T helper 1s, these are all effector cells, right? Effector cells. Except for, of course, the memory, which are memory cells. So T helper 1s activates phagocytes via interferon or IFN gamma and tumor necrosis factor alpha. Whatever I list here, these ones you should have just remember what they are. These are two different types of cytokines that are really important in helping boost the inflammatory response. But macrophages, sometimes they have intracellular pathogens that sort of just hang out there. And this Th1 response can really kind of boost this phagocytic response, but also these are pro-inflammatory cytokines. So this whole Th1 response also leads to greater inflammation. Th1 is important if you have an intercellular pathogen. So if you have something that's stuck on the inside of a cell, like Salmonella, for example, this Th1 response is actually quite beneficial because it does really boost that phagocytic response and can improve that inflammatory response too, to help clear those guys that go on the inside of the cell. If you have an extracellular pathogen, Th2 is beneficial because it boosts that antibody production in B cells. So extracellular. Extracellular pathogen. T follicular helper cells, these guys are stay inside the lymph node. They migrate to the follicles that I mentioned are the B cell zones, and they are responsible for activating the B cells with this particular chemokine called interleukin 4. So this would be, again, for an extracellular pathogen to produce that antibody response. Th17 is associated with inflammation. Oftentimes the Th17 response is associated with some really negative effects. Anyone who has autoreactivity or autoimmune disease, oftentimes you'll find an elevation of these Th17 cells in those autoimmune disorders. T regulatory cells are vital for regulating and actually like turning off our immune response. So interleukin 10 is a key cytokine mediator of regulation. So it reduces activity and constantly combats the overreactive immune system. So these T regulatory cells are really, really vital for maintenance and regulation of our immune system. So these are the general classes of different types of effector T cells, and then memory T cells. Actually, there can be memory T cells of all of these different classes, but we'll just keep it general and just say these T helper cells will form memory cells that are these long-lasting cells that can be activated at any time in the future. So let me show you a picture from your book, and then I'll kind of go back to our lymph node slide and show you where the destination for some of these guys are. This is from your book. Again, I mentioned to you that it depends upon what cytokines are released or what message is being communicated by this antigen presenting cell, what the nature of this, this CD4 cell will become, like which type of effector cell. And each activation event, so this would be activation, and then clonal expansion is listed over here. So activation will trigger this cell to turn into one of these ones. So you actually will have only one type of effector cell generated for each activated CD4 positive T cell. But which type of effector cell will depend upon this initial message that's being communicated by this antigen presenting cell. And we're not going to go into the specifics here, but essentially you can kind of think of it, whatever these guys are actually producing is usually what triggers that in the first place. So interleukin 4, if that's secreted by, and it's not usually secreted by APCs, but by other types of cells in that same environment, that will really boost the production of T follicular helper cells for this CD4 positive T cell. Interleukin or interferon gamma will push the production of T helper 1 cell that produces interferon gamma, right? So you can kind of get an idea for the signal that's required by looking at the signal that is sent out by each of these effector cells. Usually it's like the same thing. So whatever that signal that comes in from the APC, it will decide essentially the fate of this CD4 positive T cell, and this activated T cell will then expand or replicate and differentiate into one of these classes of cells that we just mentioned. In addition, no matter which type of cell is produced, there's always a subset of memory T cells that are generated during a clonal expansion process. So let's describe them a little bit more detailed, actually pretty much exactly like what we just said here, but this is a good visual image as to what's happening. So here's the effector cells that can be produced. There's T follicular helper cells that then go and can activate B cells through different types of cytokines and physical interactions with B cells at the germinal center or at the follicle to create expanding B cells and a humoral immune response. So this is the production of plasma cells that can then go and produce antibodies. Here's Th1 versus Th2. Th1 activates macrophages, also can enhance the inflammatory response. Th2 is really important in helping boost the antibody production, so they don't actually start the process, but they can go downstream and really push the production of these B cells to make more antibodies. T helper 17 is responsible for heightening the inflammatory response, and then T regulatory cells produce interleukin 2 to help reduce or dampen the immune response. So before we go into cytotoxic T cells, let's just go back to one of our visuals here. Let's go back to our lymph node. All right, so back over here in the beginning, we talked about how these antigen-presenting cells come in from the intertissue, right? They're fully loaded and they're gonna drain through the follicle or drain through the lymph node to get to the T cell zone where it's able to cross-present to both T helper cells and cytotoxic T cells. Now, all of these cells here are naive in the first place, that's they're hanging out here. And once they become activated by this antigen-presenting cell, then it depends on what the nature of the effector cell is, but they'll either stay or they will leave. So cytotoxic T cells, these guys, once they become activated, they leave. So the effector, TCs, cytotoxic T cells, leave the lymph node and migrate to tissue. And they leave through the efferent vessel, right? These CD4 positive T cells, so it depends upon what the cell is going to do. But let's focus on these T follicular helper cells. So if this becomes a TFH cell, T follicular helper cell, that means it is an effector cell if it's a follicular helper, and these are CD4 positive T cells. They will come and migrate to the B T cell zone, right on the edge of the follicle between the T zone and the B zone. So they're going to come and hang out right here and wait for B cells to come to get activated. The other types of T cells, most of them do hang out here, but some of them can leave as well. So if you have Th17, Th1, and Th2 can also leave. Some of these guys will go and become resident cells in the tissue. Others will go into circulation. So it depends upon the destination of these guys, but they'll go and leave, and they can also migrate to that injured tissue there and have their response there, or they can stay in circulation and be systemic and have their response there. So again, it kind of depends upon what's happening in the body where these guys will go, but usually the fate of all of these effector cells is that they leave, and then they home back to, or they head back to the area that they got activated in the first place. So these guys will go out in circulation and essentially get pulled back to the area that they were activated at that injured tissue spot. Memory cells usually go into circulation or can go into the tissue, same as the other stuff, and just hang out and wait for downstream, you know, for the second time around or third time around that you see those different types of antigens. But let's focus up here also. Actually, I guess we have to talk about cytotoxic T cells. So I was just mentioning the migration pattern of these guys. So most of these effector cells will leave and then go out to tissue. Some of them, specifically the T follicular helper cells, will stay inside the lymph node and actually be responsible for downstream activation. So let's look at cytotoxic T cells from the sides. Those were T follicular or T helper cells, but cytotoxic T cells, these ones differently. Once they become activated and they expand and they become effector cells, let's just clarify that real quick. Once they not just activated, but become effector cells, they leave the lymph node and they migrate to injured tissue. All right, so at the injured tissue, they will then go and try to find a perfect match, right? So here they're gonna look for infected cells. So now we're looking for infected self cells presenting MHC class 1, presenting foreign antigen. If the same foreign antigen is the one that activated these guys in the first place, then you'll have that, then you'll have CD8, of course, binding MHC class 2, and the foreign antigen bind to T cell receptor, right? If this is the same foreign antigen as before. If this interaction occurs, this will then lead to effector function. Leads to and what the function of these effector cytotoxic T cells is essentially to cause apoptosis of any infected cell through the secretion of these two major chemicals, perforins and granzymes. Perforins are responsible for perforating or penetrating the holes, the membrane with holes to make them more porous, and then granzymes are secreted right afterwards to get into those porous holes and attack all the different proteins. This usually leads to such damage inside the cell that the cell commits apoptosis, which is like programmed cell death or cell suicide. This leads to the destruction of that cell. This can be viral infected cells or cells infected with any intracellular pathogen, cancerous or abnormal cells, and also any foreign cells come in. This is in part why graft rejection is such a big thing and why it's important to dampen the immune system altogether for people who have received a graft so that they do not get their tissue rejected by our cytotoxic T cells. Here's a picture of what this looks like. Again, remember that this is in the lymph node that you have initially cross-presentation of the antigen presenting cell with MHC class 1 that's presenting foreign antigen, and the ability for the CD8 positive T cell to have T cell receptor engagement with that foreign antigen and a co-receptor recognizing MHC class 1. In addition, I've already mentioned that this activation is not quite enough. In order for clonal expansion to occur, that T helper 1 neighboring activated T helper 1 cell that's now secreting interleukin 2 must have interleukin 2 secreted, binds to interleukin 2 receptor on that CD8 positive activated cell to become an effector cell. So this is clonal expansion here, this process where memory cells are always made as a small subset, and then the effector cells are produced that then go out into circulation and go back into the tissue that is that has the injured tissue. Here's a picture of an infected host cell, so like a viral infected or bacterial infected host cell. These guys, remember, are producing MHC class 1 constantly, and if they're infected, they're going to be presenting that foreign antigen on the surface of MHC class 1. If that antigen is a perfect match, excuse me, if that antigen is a perfect match to this T cell receptor from way back in the beginning, then this leads to the effector function. This will engage this cytotoxic T cell that's an effector cell to degranulate and produce perforins that perforate the membrane and create holes in the membrane, and then granzymes, which are these enzymes that come in and really destroy the proteins that are inside the cell, and this will lead to programmed cell death or apoptosis, right? So here's that destruction of host cell. I don't think I want to do this one. Let me see if I can get a new. No. I wish there was a way that I could get another page. Okay. Okay, I could just draw it on here. Let me come and erase these guys here for just a second. Let me just draw that for you real quick. So these cytotoxic T cells, once they leave and they migrate out into tissue, let's just draw that out just as a reminder. I know I drew this from in the last set of slides, but let's just draw that out here. So in the tissue, let's say we have our own self cells. And that means that these guys, if they're self, they have MHC class 1. And if you recall from our last set of slides that we talked about, these MHC class 1s are constantly producing self antigen, right? But if this is infected, let's say that means it has a viral infection, then each of these little virus parts are going to be presented on the surface of these self-infected cells, right? So this would then be foreign antigen being presented on the surface of self MHC class 1. And here is our now effector. Cytotoxic T cell, these are still CD8 positive T cells. They still have their same T cell receptor, and this is important. They have the exact same receptor as they had in the first place. These guys are now in tissue and and they're looking for the same exact antigen that they were activated with in the first place. So here's the T cell receptor, that perfect interaction there. And these are CD8 positive T cells. That means they still have their CD8 co-receptor. And that means they're looking for MHC class 1. And now that we're out in tissue, this whole cross presentation isn't working anymore. So anytime this receptor engagement occurs, where you have MHC class 1 being recognized by CD8, T cell receptor binding perfectly with a foreign antigen. Now this will then lead to effector function, okay? So here, because this is an effector cell, this will release to effector function, which is degranulation. I can't fit that in there. So this leads to effector function, which is degranulation and the release of perforins and granzymes. So perforins will penetrate holes in the membrane here, and granzymes will come in and just completely destroy or degrade many of the proteins that are there, and this then leads to apoptosis. Equals death. Okay, so this is all out in the tissue. That's what I'm trying to say, not in the lymph node, in the tissue. Okay, so that's only these effector cytotoxic T cells. When they're out in the tissue, this is what they do. When this receptor engagement occurs, it will cause the degranulation of these guys, the release of perforin and granzymes, and these will perforate the cell and then degrade the proteins inside that cell, which will then lead to apoptosis or cell death. So in addition to these CD8 positive cytotoxic T cells, there is another class of cells called natural killer cells. And these ones are those strange cells that I mentioned to you, are kind of part of both the innate and the adaptive immune system. These guys act like these cytotoxic T cells that we just mentioned, but they have no antigen specificity. Therefore, because of this one fact alone, they're technically part of our innate line of defense, but due to their nature, their reactivity, they are mostly considered part of our adaptive line. So they kind of have this strange crossover. Their job is to essentially do exactly what cytotoxic T cells do, except instead of binding to and recognizing MHC class 1, they bind to and recognize any cell that has no MHC whatsoever. In fact, we call them natural killer because their inherent nature is to go around and kill every single cell. And the only reason they don't kill your own cells is because you have MHC class 1 as an inhibitor. So the MHC class 1 is required to prevent the destruction of our own cells. So let me kind of show you how this whole concept works. So here we have on the side, here is a self cell that is infected with pathogen. Now, oh, that's interesting. So here's an intracellular pathogen. It is intra, not inter. An intracellular pathogen. So under most normal circumstances, this pathogen, as it's infecting the cell, will also get processed by the MHC class 1 machinery inside the cell, and you'll have the presentation of a pathogenic antigen on MHC class 1. This, as just mentioned, will lead to the activation of our cytotoxic T cells and induction of apoptosis through the release of perforins and granzymes. But as just mentioned also, these natural killer cells, they're naturally killer. They want to just go and destroy everything, secrete those perforins and granzymes, but they are inhibited by MHC class 1 receptor. That's even on an infected cell. Even with self cells, any MHC class 1 receptor will inhibit the activity of natural killer cells. Now, there are certain circumstances where either through cancer or through particular pathogen reactivity, the production of the actual MHC class 1 presenting molecule is depleted. So in this case, you have a cell that's infected or cancerous or abnormal, and it has no MHC class 1 presentation on it at all. Therefore, cytotoxic T cells cannot work in this regard because there's no presentation of antigen, right? So cytotoxic T cells would be out of luck. This is where we have our natural killer cells being so important. They will then recognize, well, not recognize, but they will then detect the absence of MHC, and then they will cause destruction of that cell, right? So cytotoxic T cells and natural killer cells are very similar in their effector function. But natural killer cells will essentially recognize cells that are missing all their MHC class 1 receptors and destroy them, versus cytotoxic T cells, they recognize MHC class 1 that's presenting foreign antigen and cause the destruction of that cell. So these questions I think are helpful for you to just kind of think about the overall function of our cell-mediated immune system. The first question: during development of T cells, all naive T cells undergo a negative selection, whereby if they bind to presented self-antigens, they're signaled to self-destruct. Why is this important? I'm sure you can figure that out. The second question: come up with an analogy for the role of an activated T helper cell. Now, because of their central role in essentially activating everything else, including producing all of the effector cells that then can go and activate B cells, or producing interleukin-2 that then can go and activate cytotoxic T cells, one of my students came up with the analogy that these T helper cells act like 911 operators, where whatever the call is, the nature of the call, which is like the dendritic cells, like telling the body what's wrong with it, that particular call will then trigger some kind of downstream effector function, right? So the 911 operator receives a call and then chooses what type of response is needed, which is very, very true of these T helper cells. Maybe that'll help you. And then this third question here: HIV infects CD4 positive T helper cells. Explain the effect on the immune system. Well, now that we've seen the vital role of T helper cells, hopefully now you can see why HIV infection is so damaging and causes all like systemic autoimmune disorders, or decreased, that's not the word, overall, what's the word I'm looking for? Reduction of your immune system, immunocompromised, thank you. Immunocompromised position, because not only does this infection wipe out just those CD4 positive T cells, but we again just talked about how important they are in downstream activation of both B cells and your cytotoxic T cells. So the absence of your CD4 positive T helper cells really makes an entire huge cascade reaction on the activation of everything else in your adaptive immune system. So with that information, we can now move on to B cell activation. And let's go back to our chart. I'm going to start to erase some things here. Okay. So let's head back over here to our lymph node. We just talked through the whole production or activation and effector function of our CD4 positive and our CD8 positive T cells. And this is where we're going to focus now, this one subclass where the CD4 positive T cell becomes a T follicular helper cell and migrates over here to the B and T cell zone. So I'm going to erase some stuff at this point. And I'll redraw, I think I can erase everything at this point, and then I'll just redraw what's happening at that B and T cell zone. Okay. And before we get there, let's redraw our B cells, and let's remind ourselves what B cells look like. Okay, here, let's draw it over. Let's just try it here, and then I'll just redraw this whole thing. B cell. All right, B cells, when they leave the bone marrow, they are naive, right? They have never seen antigen. They're going to go through circulation, and they're going to end up at the secondary lymphoid organ, just like the T cells. Now, these guys don't go through the T cell zone. They go directly to the follicles, because there's circulation that actually deposits them right into the follicles. Now, B cells have a couple of important receptors. They have a lot of things on the surface, but two, very importantly, that we'll talk about here. They have their B cell receptor, which, if you can recall from our last chapter, or from our last set of slides, are immunoglobulins, and they're, whoops, I wrote that one wrong. These are immunoglobulins, and they're class D. So a B cell receptor is immunoglobulin class D, almost always. Or we can say IgD positive cells. That means that they're expressing IgD. Immunoglobulins have that FAB region that has the variable region that's important for antigen binding. and the constant region, which is just of this particular class here, and a transmembrane region that anchors this immunoglobulin into the cell surface. In addition, these guys also have MHC class 2. And where do we see MHC class 2? Only on antigen presenting cell. So they have MHC class 2. Okay, B cells, sorry about the dog. B cells, sorry, I'll write naive. Naive B cells must come in contact with antigen. This can happen in the blood during migration to the lymph node. This can happen in the lymph node, in the follicle, with foreign antigen in the lymph, right, coming in from injured tissue. This can happen through an antigen presenting cell, presenting antigen to the B cells in the lymph node or anywhere else actually. So it can be through APCs, but this we're not really going to focus on very much. So this interaction with antigen can happen either in circulation or in the lymph node, but somehow, some way, this B cell comes in contact with an antigen. It can be the whole pathogen in the blood, right? If it's a virus or intracellular or extracellular bacteria that got into the blood. But no matter what, no matter where this thing happens, this comes in contact with is highly specific. Because this is through the immunoglobulin receptor, right? The immunoglobulin FAB region. So somewhere, somehow, and let's just pretend like this is a virus that's in the blood. And most of the time, it's the glycoprotein spikes that actually have that reactivity with the antigen or with that antibody. So somewhere, somehow, this B cell, this naive B cell, comes in contact with an antigen. And in our example, we're just going to say this is in the blood and this virus interacted with this B cell. And it's through this highly specific interaction between the immunoglobulin receptor, FAB region, and the foreign antigen, and specifically the epitope on that foreign antigen, if you guys remember that. So this then will cause this B cell to undergo the process of phagocytosis. And migration to the follicle, B T cell zone, in the lymph node or in the secondary lymphoid organ. So this guy, this whole engagement here will lead to phagocytosis, right? Which of course is the engulfment of this whole thing, the processing, the digestion of this stuff. And then remember, at the end of the phagocytosis, this guy now becomes an antigen presenting cell and presents foreign antigen, phagocytosis, migration to the B and T cell zone, and presentation of foreign antigen on MHC class 2 only. This does not cross-present, does not cross-present. Now, the cool thing about B cells is that even though this requires a highly specific receptor engagement here, the process of phagocytosis means that whatever this thing is, if it's a whole virion particle, the whole thing will get digested, just as we talked about with macrophages and dendritic cells. And as an antigen presenting cell, this will present not just the little epitope that caused the activation of this cell, but anything, right? All the different types of antigens it can extract out of this pathogen or the molecules that it digested. It's going to present a whole bunch of foreign antigens on the surface of its membrane through MHC class II. And this becomes important because in order for this B cell to become an effector cell, to undergo clonal expansion, it actually needs to engage with a T helper cell, a T follicular helper cell, at this B and T cell zone. And this is a specific interaction. So number 4 is at that B and T cell zone. So it's going to wait for the binding of a T follicular helper cell. Let's draw another one of these receptors down here. So let's say that this is that B and T cell zone, right? And here on the other side, if this is the follicle where the B cells are, this is the T cell zone. And we have an effector, T follicular helper. These are effector cells. They have already undergone clonal expansion from their original CD4 positive state, right? So now they're CD4 positive T follicular helper cell. These guys have a T cell receptor, if you recall. So now at this B and T cell zone, this B cell needs to await further activation by a CD4 positive T follicular helper cell that has a perfect T cell receptor interaction with that presenting peptide from an MHC class II molecule of that B cell. Again, it's cool that this B cell, can produce a whole bunch of different antigens from that one interaction there because of the process of phagocytosis. So even though this interaction must be specific, at least it doesn't have to worry about being as specific as it was in the first place. So it doesn't require the exact same antigen that activated this B cell to cause this interaction here. It could be any kind of peptide fragment from this molecule. As long as this receptor engagement was perfect, this will lead to phagocytosis, and phagocytosis just breaks apart that whole thing, that whole, let's just say, virus particle. And as an antigen presenting cell, everything, or a whole bunch of different types of antigens, have been presented on the surface of this cell. So here is this T cell receptor. If this is a perfect match here, this is still a CD4 positive T cell. I don't remember what my colors were, but whatever. So here's that CD4 positive or CD4 co-receptor, that binds and recognizes MHC class 1, or class 2, I'm sorry. Now, just as we talked about the last time, now that this is an effector cell, this is enough to activate this one. And this causes the release of interleukin 4. IL-4 receptor. And interleukin 4 can then go and cause clonal expansion. So it turns it from a naive cell into an effector cell. So this will be clonal expansion. Okay. And clonal expansion for B cells, let's write down our checklist here. Presentation of foreign antigen on MHC class 2. So the activation, or Um, clonal expansion. So clonal expansion produces the effector cells, which are called plasma cells, which will then produce antibodies, secrete. This also will produce memory cells and also can generate regulatory cells. But this is by far the biggest category of cells that are produced under this clonally expanded B cell, and then we'll talk about the function of these. Let's go back and look at our slides. Okay, and these might have a slightly different sequence, but let's look at. So for B cell activation, first a foreign antigen bind a naive B cell on the immunoglobulin receptor, or IgD, cell surface molecule, and this is a highly specific interaction. This receptor engagement leads to phagocytosis, so antigen processing and presentation on MHC class II, and the migration of these B cells to the B and T cell zone in the follicles. Here, that B cell will then await a T follicular helper cell to come and recognize that same exact antigen, bind, and then secrete that interleukin 4. So if interleukin 4 is released by T follicular helper cell, that B cell will then receive that signal and become undergo clonal expansion. And I would actually argue that activation goes up here, sorry. This is an older slide. Let me put activation. B cell activation is actually when it comes in contact with antigen, it's been activated. And then clonal expansion is definitely what happens from that interleukin signal. Clonal expansion then leads to that effector cells, plasma cells, and then memory cells, right? So the effector cells would produce antibodies and secrete them out. And then the memory cells are long lasting. The, again, important thing to note is that these antibodies that are made and secreted out of the cell have the exact same antigen specificity as this immunoglobulin receptor that bound to that antigen in the first place. This is a picture from your book. Let's just look through it real quick. I know it says kind of small, but you can read it on your own time. But here is that organism, and as mentioned, that binding of the antigen can be in many different places. This can happen in the blood and circulation. This can happen actually as part of that antigen presenting cell inside the lymph nodes. This can happen in the lymph nodes when a microbe comes and filters through the sinuses from the draining lymph. So there's lots of different ways that a B cell can become engaged with this foreign antigen, but let's just do this as an example where there's like a virus in the blood, and as that naive B cell is heading towards the secondary lymphoid organ, it comes in contact with the virus, and if this immunoglobulin B cell receptor on the surface of the cell has perfect specificity to some part of that microbe, then this will lead to activation of this B cell. And these guys will then become phagocytic cells, right? So they're going to phagocytose that particle, presented on MHC class II, and also migrate to the B and T cell zone, where T follicular helper cells, these effector cells, may be waiting to test out that perfect match. So if indeed this B cell acting as an antigen presenting cell is presenting foreign antigen on MHC class II, and that foreign antigen is a perfect match for a T follicular helper cell that's hanging out at that B and T cell zone, then you can have this whole receptor engagement, and this leads to two different things. It does lead to crosstalk between this B cell and the T follicular helper cell, but most importantly, interleukin 4 is secreted by this T follicular helper cell to help promote the expansion of this presenting B cell. So this would be B cell clonal expansion. I know it says activated, but I don't really like that too much. It's really, this leads to B cell expansion. So here is now B cell clonal expansion, where you now have effector cells. So here's the effector cell, which is a plasma cell that can then go and produce antibodies. Here's memory B cells that can expand, but then they just stay in this partially differentiated state. And then regulatory B cells that are also important in helping regulate the immune system. All right, now we can look at the function of those antibody classes. So here is a quick reminder of what these antibodies look like, with the 2 heavy chains here on the inside, the 2 light chains here on the outside, the designation of the variable region here with the antigen binding cleft, or the region that functions in antigen binding, FAB region, or the functional constant region here down at the bottom. And this is what gives the class designations. So the functions of antibodies, there's a whole bunch of different things that they can do, but we'll look at 4 in particular that are really, really important. The first here is a neutralization. So antibodies will bind to and neutralize. Actually, you can kind of see it in both ways. Yeah, I don't know why they separate this thing out, but neutralization is essentially where antibodies are binding to the surface receptors of viruses or even of bacteria to neutralize their activity, so they can no longer bind to the receptors of host cells. So this would be neutralization. Opsonization is where the coating of a bacteria or virus leads this, the FC tail, that constant region tail just kind of hanging out. And that FC tail can bind to FC receptors on the surface of macrophages, and it really enhances phagocytic activity. So this is called opsonization, is where coating enhances phagocytosis from phagocytic cells, in particular from macrophages, and that's through binding of the FC receptor to the FC tail portion. Agglutination down here is where it's really a major function of the FAB and the hinge region here, so the flexibility of these 2 different arms of each of these antibody molecules. So you can have one arm binding to one pathogen and another arm binding to a different one. And this cross-linking of various pathogens together is called agglutination. You can imagine it as like gluing these guys together. And this creates these things called immune complexes that are really easy for the body to get rid of, either through phagocytosis, or if this is out in a mucosal secretion like your gut, then it really is easy for your body to clear this through gut motility. This concept here doesn't happen very often. This is complement fixation. So the binding of antibodies to bacteria can lead to the activation of the complement cascade. That is true, and that is certainly a major player in inflammation. But what you're seeing here, the perforation of these cells, that doesn't happen very often. Nonetheless, complement cascade can get activated through antibody-antigen interactions here. Antitoxins, I would just imagine this is still another concept of neutralization. So neutralization really is one of the major functions here. It can prevent toxins from binding to surface receptors, can prevent viruses from binding to surface receptors, and can prevent bacteria from being able to bind to the surface of a bacterial cell or of a host cell. So I'm gonna add to this. What I would normally do is write all this on a whiteboard, but I don't wanna write all this stuff out again since we have it here. So let me add to this just a little bit about which particular antibodies are great at doing this. If you recall, we have these 5 different classes. So we have immunoglobulin class D, which are usually B cell receptors. We have immunoglobulin class E. I'll just do the 2 that are in lowest abundance. And E is usually involved in the allergic response with mast cells. So as far as in circulation, IgD and IgE are almost never found, or very, very low abundance, because usually they are either bound to the surface of B cells as B cell receptors here, or for IgE, they're involved in the allergy response. You can measure an IgE titer if you're trying to determine if someone is experiencing an allergic response to something. So if you've ever gone in to do, to test for that one, that's what they're looking for. But otherwise, the three major classes of immunoglobulin molecules that are responsible for pretty much all of these functions here are IgG, which is the most abundant, and long-lasting. IgM, and let me just say, these three exist as monomers. Means it's just one molecule, just like this. IgM is the first class produced during the first time around, during your first response. And this can be mucosal, which means it can be secreted. And these guys always exist as a pentamer. Penta means 5, right? 5 of them. And then IgA, this usually exists as a dimer. Dimer is 2. And these guys are most abundant in mucosal response. All right, we'll look at those again in just a second, but just to get them on the chart here. So as far as each of these FC or each of these antibody functions, let me just kind of mention which cells are really great at doing, or which classes are really great at doing each of these things. So if you recall also, let me just mention, this is the FC tail, and this is the FAB portion. Okay, so as we look at each of these activities of antibodies, consider what portion of the antibody is important in generating this interaction. So this first one, opsonization. Let me change my color again. For opsonization, macrophages have something called FC receptors, or FCR, that is important in, of course, binding the FC tail of these ones. And if you consider these different classes, first of all, IgE and IgD are not really in circulation. IgG is the only one that exists as a monomer that has the ability for this FC tail to stick out. So IgG is a really important class in opsonization to make that FC tail available for binding to the FC receptor. But IgM can do this too. I would say IgG is the biggest one, right? The most important class in this concept of opsonization, where opsonization is coding to enhance phagocytosis or phagocytic action. For neutralization, all antibodies can do this. IgG is great at this, IgM is great at this, and IgA, so all of these classes that are our highest responders, IgG, IgM, IgA, all of them can do this. And the portion of the antibody that's most important in this response is the FC portion. I'm sorry, FAB portion. If you look at it, FC tail doesn't really do anything. It doesn't matter which class of molecule you have on here, because it's really just binding with high specificity the FAB region to that surface protein. Here for agglutination, now this one, it's about cross-linking these different cells or viruses. IgG can do this, it's okay. But these pentamers and the dimers are fantastic at this, right? So IgM is ideal because it has 10 binding sites with 5 of these molecules stuck together. So IgM is fantastic at doing this. IgA is great at this too, because it's a dimer, right? So there's 4 different FAB regions to cross-link all of these guys together. For complement fixation, it's almost exclusively IgG, although IgM can do this too, can weakly activate complement, but mostly it's IgG. And then antitoxin is basically the same as neutralization, right? So here's the 4 big functions of antibodies: opsonization, neutralization, agglutination, and complement fixation, and the major classes of antibodies that are great at each of these things. And so if you also just think about what's our most abundant immunoglobulin response, IgG. IgG is important in all of these activities, right? IgG is kind of okay at agglutination, which perhaps is why IgM is the first antibody that's produced during an adaptive immune response. IgA is really great at this, and this is all in mucosal too. So these classes, this is a table from your textbook, just a nice picture of each of these things. Also note that these little blue dots on here are glycans, those little sugar molecules, and you can see how important these glycans are in providing some of the structural framework of each of these molecules. In fact, there are many studies showing that if you take away these glycans, then the entire molecule loses its function or alters its function altogether. This is the one that I studied for my dissertation, this IgA, and even though there's no blue dots on this, I'll tell you, this is one of the most heavily glycosylated molecules, because both this green component here called the secretory component and that dimeric molecule are coated with over 13 different glycan molecules. So this is showing you a few different features of each of these molecules, what their size is, what their percentage is in the serum, their half-life, so how long they last in the serum, whether they are important in utero protection of the fetus, which only IgG is, complement fixation. The Fc portion can bind to these different things. So really it's just IgG that is important in that opsonization to enhance phagocytic action. And then here's a description of their biological function. This is a concept of the first and second time around. So the first time you're exposed to a pathogen, a virus or a bacteria, we recall this as our time frame we put behind in the early part of chapter 12, where your first exposure to antigen causes an inflammatory response. Right? So right away you have an inflammatory response that takes seconds, minutes, maybe hours, and then you have those late responders, dendritic cells and macrophages, that pick up antigen and head to the lymph node, and then they try to search for that perfect match of T helper cell. Usually this latency is because of that amount of time it takes to find that perfect interaction between that antigen presenting cell and the T helper cell. Once that T helper cell is engaged and begins its expansion and then produces those effector cells that can then go and activate B cells, etc., right? That's when you have our response actually start to take off. So this whole latency period can be days or potentially weeks, but usually it's days before you get that first exposure. That first response. So once you do have that response, then look at this little chart. So we have IgM in red and IgG in blue. You initially have an IgM response that actually produces quite a bit of antibodies right away. And then we call this seroconversion, is the switch to IgG. And this is a longer lasting response. And in fact, IgG levels can remain high for a long time after the pathogen is actually cleared. So you can even detect IgG levels in circulation for sometimes years or longer after that first exposure. In fact, just to give you guys some context, the serology testing that's being done right now or being produced for coronavirus actually differentiates between IgM response and IgG. And the concept here is that if you have an IgM level pretty high in your body that's detectable, that means that you're actively fighting off that infection. That means that you are infected. But if you have an IgG response, usually that means that you are either in convalescence or recovered completely, right? So the late stages of infection or recovery where you still have detectable antibody levels. Now, after, eventually, you'll have lower antibody titers. Usually this does not go down to nothing, but it goes pretty low. There's no virus or pathogen in your body anymore, right? And you undergo however long where you don't see that antigen again. But anytime you have another exposure to antigen, this is called your secondary response. But here, remember, you've developed memory cells the first time around. So those memory cells are what are going to be activated instantaneously. So within minutes to hours of that second time around, you'll develop an amnestic response or memory response, where here you have a giant production of IgG with a low or late production of IgM to produce even higher levels of antibody than the first time around. Right, so this is primary versus secondary response. It's just the first time you see a pathogen versus the second time you see it. And secondary, it just means any subsequent time. So you can become exposed to that same organism over and over again, and each time you'll develop the secondary response. All right, and that's it for B and T cell responses. So we looked through the whole process of B and T cell activation and then effector function. Let's just cover immunity here, and then we might just pause and do vaccines in a different set of slides. So for immunity, there's really just a few terms that you need to understand, and this becomes relevant for today's day with coronavirus and what's being pushed for the development of an immune response. So a natural active immune response. These are two terms, natural and active. Natural means that this is a naturally occurring pathogenic infection instead of artificial, which would be stimulated by a vaccine or some genetically engineered pathogen. So a natural infection or a natural immune response means you have some kind of natural infection. Active means that your body is actively mounting an immune response, right? So this is a classic immune response that you get under the course of an infection. This develops a memory response, which is really important. That's how we gain the ability to fight off things for a long period of time, is through that memory response. This we know takes several days to develop, but it is a very long-lasting response. This is in contrast to a natural passive immune response. Natural passive is primarily what we're talking about is a mother's immune response, where she has at some point in her life gained natural immunity, right? Natural active immunity. And she passively transfers immunoglobulins either across the placenta during in utero or through milk for the duration of breastfeeding, right? So this provides passive immunity to the infant, but it's natural immunity because this was developed by a natural infection in the mother, but it's passively transferring that immune response, specifically the antibodies from the mom to the baby. Artificial immunity is where some medical intervention produces some type of immune response. You can have artificial active immune response where you have a vaccine, right, vaccination. And this is with an artificial, genetically engineered or modified pathogen that still produces a natural immune response. So you still can get that memory response, which is really what you're trying to boost for vaccination. Immunotherapy is what is being done right now with convalescent serum with coronavirus, where antibodies are taken from human serum and provided passively to a recipient to develop that immune response. So this would be artificial immunity. And then vaccination, as mentioned, we can cover this. Actually, let's just cover it now because we are talking about this right now with the coronavirus epidemic. So the concept of a vaccine response, the whole premise here is to stimulate your primary immune response and to prime it for any subsequent response, right? So it's a priming of the immune system for any future exposure, essentially to gain that memory response so that in the future event of exposure, you have an amnestic secondary response, which means that you're fighting it off before it has a chance to establish itself. Usually what's done here is antigenic materials, so a vaccine that's been, or a virus that's been attenuated, which means it's been modified so it's not virulent anymore, or pieces or fragments of a pathogen that are not infectious, but they provide the body the antigen to provide an appropriate response. The challenge, of course, with vaccine production is in antigen selection. We've talked so much about the requirements for specificity for that particular antigen to stimulate that appropriate immune response. It's especially vital when we're dealing with something like a virus, that only particular regions on that virus are important for binding. And so you need to have an appropriate neutralization antibody in order to get binding and neutralization capacity. So antigen selection is one of the biggest limitations for vaccine development. Effectiveness, ease of administration, safety, and cost, these are just things to consider, and some of the challenges of developing appropriate vaccine. These are some of the different preparations I just mentioned. So really common is a whole killed cell. This is, of course, for bacteria. Just take the entire pathogen and kill it through heat or through chemicals, or take a virus and inactivate it in a similar way. And that way you just have all the same molecules that the normal pathogen would have. It's just that this would not be a live organism, so it does not cause an active infection. Different types of preparation would be live attenuated virus or cell. That means that you're genetically modifying these things to be attenuated or less virulent. They can still come in and cause a low-grade infection, still mount an appropriate immune response, but they're not going to damage the cells like their wild-type counterpart does. Antigenic molecules, this is what's being done usually right now for coronavirus, is just taking the spike protein, the glycoprotein, and just using that protein itself to try to stimulate an immune response instead of putting the whole virus particle in, or genetically engineered microbes and antigens, which is another approach altogether. I think that I'm going to let you guys go through these slides. So these are just looking at all the different ways you can prepare these vaccines. So there is a risk for side effects. Almost always vaccines will elevate temperature. They'll cause inflammation at that site of injection. Some people do mount an allergic response, not to the actual vaccine, but usually to the preservatives that are required to make this shelf stable. And fever is really, really common here. So these are really common side effects for, or these localized reactions. Very rarely, so 1 in 200,000 people might develop some type of adverse event. But of course, there is this constant controversy, mostly in small subsets of the community, where the risk of getting the disease and the risk associated with that vaccine are really put into consideration. So always, and this is true actually not just with the anti-vaxxers, but this is true even in the development of a vaccine, that the vaccines must be safe and effective, and sometimes safety is there is some consideration to what is permitted for adverse effects in the human. This is usually under normal. This is why vaccines usually take 5 to 10 years to come onto the market, because of the safety risks. Even if you get a highly effective vaccine, if it causes some type of damage to the host, then of course that can't be put on the market in high abundance. But under extenuating circumstances, like with H1N1 and this current coronavirus epidemic or pandemic, then safety is important, but adverse events are acceptable to some degree when trying to push for the production of these very quick vaccines. Because the concept here is to develop herd immunity. Many of you have heard of this concept in the news, and I just wanted to mention it here. So these images give you a good idea of what herd immunity is. In this first picture here, this is a great picture of the entire world at the beginning of the coronavirus infection, where no one is immunized and no one is immune. So everyone is potentially susceptible to this particular pathogen. So when something like this enters the population, which is exactly where the world is right now, a large percentage of the individuals have the chance of acquiring that infection and getting sick from it. If we have immunization, or if people have already been exposed the first time, in either case, that you get some people who are now not susceptible to developing this disease, either because they've had it before or because they have been vaccinated with that disease. In this case, you'll still, if people get sick with that disease-causing agent, it'll still spread pretty wide in that population. Really, it's only until a great percentage of the population are either immune through exposure or immune through vaccination that then if people get infected with that pathogen in that population, it has a very low risk of infecting other individuals who are still susceptible. This is what herd immunity is trying to get the whole population immunized in high enough numbers that even if sick people come into that population, the risk of them passing on that infectious agent to people who are susceptible is very, very low. And the number here is something about 60 to 70%. So if 60 to 70% of any given population is either immune through exposure or immune through vaccination, then the likelihood that people coming into the community who are sick are going to infect the remaining susceptible individuals becomes very low. So this is where we're trying to get to with, obviously, with vaccination for all of the childhood illnesses in our population right now, like whooping cough or measles or rubella, etc. But also in current outbreaks right now, like with coronavirus, this is where some communities, like in Sweden, have thought to allow the disease to progress so that people in the community get sick with it and become naturally immune, active natural immunity, so that they develop this herd immunity so that in the next season, when this virus hits again, that less of the population will be impacted. And that's where we're going to leave it here today.